This is awesome.
So we left off, take out your notes please, on the physiology. So we talked about sort of general osmosis and diffusion and how that can affect cells and how it works in general. And now we talk a little bit about specifically in the cell, how do substances move back and forth? What allows that to happen? What structures are there that allow for molecules to travel through that plasma membrane? So you guys described to me in our question today sort of the structure of the cell membrane. And that structure was actually not really described well until the 1970s. So in the 1970s, some scientists put together what they call the fluid mosaic model of the cell membrane. It's a term that you probably will see. And this fluid mosaic model basically describes the cell membrane as this sort of phospholipid bilayer with all these different substances embedded inside of it. Okay, that's where the mosaic part of that comes from. And we'll look at that a little more closely in a minute. But when we talk about, in living things, the transport of materials, we can talk about intercellular transport or intracellular transport. So again, those two prefixes. Inter meaning what?
Inside.
No.
Outside.
No.
Between cells.
Between. Inter means between two things. Intra, nope, means within one thing. So if we're talking about intercellular transport, we're talking about the movement of things between cells, or between a cell and the external environment. Okay, so diffusion is one way of molecules moving, of intercellular transport. Oxygen can diffuse from the air in the alveoli into the red blood cells. That's intercellular, between the cell and the external environment. So the cell membrane is a phospholipid bilayer, like we talked about. It's a double layer of lipids, okay, with proteins embedded within that bilayer. The external proteins often have carbohydrates attached to them that have various functions. And these proteins do different things. So we'll talk about some of those. Because some of these, especially the ones with carbohydrates attached, are what we call receptor molecules. And we'll refer to these receptor molecules when we talk about the immune system, when we talk about the endocrine system, because hormones work by attaching. A hormone molecule attaches to a receptor molecule that's on the outside of the cell membrane and signals it, tells it to do something, okay, causes some change to happen. Okay, and that's what some of those molecules do. If the cell membrane is impermeable to a substance, okay, there are various ways that those molecules can get into the cell. Even though they cannot pass through the cell membrane, they can be ingested into the cell by pinocytosis or phagocytosis, which are really just variations of the same thing, of the cell membrane changing shape to take in molecules from the outside. Okay. So here we see a cell, we see the tiny, very thin cell membrane around the outside. When we talk these means of taking in material, phagocytosis is when pseudopods of the cytoplasm ooze around the food molecule or ooze around whatever is being ingested, eventually connecting, and then that particle is then inside of the cell in a small sort of vacuole. Pinocytosis is basically the same sort of thing, but it's a pinching in. So as the membrane pinches in rather than surrounding the cell. This is called endocytosis. Again, cyto meaning cell, endo meaning inside. There's also a type of endocytosis called mediated, okay? Receptor-mediated endocytosis, where the penocytosis will only begin after certain molecules bind to receptors on the cell membrane, and that signals the cell to start to pinch in and form a vesicle inside. If we talk about moving things in more complex organisms from cell to cell, the cellular level happens via diffusion, but in larger organisms, there are different types of vascular tissue. Arteries, veins, capillaries, which we find in animals, that can transport things from one area to another. In plants, we have the xylem and the phloem, vascular tissue, vascular bundles, okay, that can carry material from one part to another. So if we look here, this is showing a cross-section of a leaf. You have the leaf, top surface of the leaf, the bottom surface of the leaf, then you have cells in between, and things like carbon dioxide. It can diffuse into the leaf, and there's air around those cells. There's water and extracellular fluid around them, and so this material, this gas, for example, can diffuse through that material from cell to cell. Inside of cells, there is the process of cyclosis, which we talked about, which is the streaming of cytoplasm, okay? Where basically the cytoplasm can move, controlled by the cytoskeleton. And you can see in this video here, what are these green circles? Chloroplasts. Yeah, chloroplasts. And you can see them moving around through the cell as the cytoplasm moves. Okay. Also within a cell, the ER can transport things through that system of membranes from one part of the cell to another. Molecules can diffuse in the cytoplasm from one area to another. So these are all ways of moving things within a single cell. So ER moves things through membranes. Diffusion just moves materials from a higher concentration to lower concentration in the cytoplasm. So when the cell itself absorbs material from the outside, okay, that's these materials getting from the outside of the cell, which we call the extracellular fluid, is the stuff around cells, through the cell membrane and actually into the interior, into the cytoplasm of the cell. So here we have, again, the fluid mosaic model of the cell membrane. You can see here the phospholipids, 2 layers. Phosphorus heads facing out and facing in, and then all the tails lined up. Okay? Embedded in the cell membrane are other molecules, cholesterol molecules, proteins, like we said, transport proteins, receptor proteins, all sorts of different things that allow for various cell functions. Okay. You can see the glycoproteins have those carbohydrates attached to them. You have the cytoskeleton, which attaches there. Various types of proteins and receptors and recognition proteins. So things get into the cell in two ways. One way is called passive transport, one is active transport. Passive transport, by definition, does not require the expending of energy for molecules to move. So when we talk about oxygen diffusing into the cell, that's a form of passive transport. It just happens on its own. There's no energy required because the molecules are moving down the concentration gradient from higher to low concentration. And it does this due to just sort of random energy of moving molecules. And the kinetic energy of molecules bumping into each other and moving causes molecules to go from higher to lower concentration. Osmosis is a form of passive transport. The water, and there's no energy being added here, but due to osmosis, water is pushing that molasses up this tube. Active transport does require energy. Active transport can move substances up the concentration gradient in the opposite way that they normally would go, from a lower concentration to a higher concentration. There are times when cell needs to move molecules the opposite, and that takes some energy. Also, moving material, getting material into the cell through penocytosis, phagocytosis, moving things within the cell by cyclosis, they all require energy. To move the cell membrane, to engulf something, or to move the cytoplasm, that requires some energy. We said that only certain types of molecules can diffuse through the membrane. Generally non-polar substances, carbon dioxide, oxygen, small molecules, just kind of pass through the cell membrane as if it's not there. They diffuse very easily. But for most molecules, they are not able to get through. Okay? Polar molecules, larger molecules can't fit through. Polar molecules that have a charge to them, they are not able to go through because of this central layer, because of this hydrophobic layer of lipids on the inside. So they need a means of getting through. And the means they have of getting through is through these proteins that are in the cell membrane. Okay? These transport proteins. Okay? These transport proteins work in some ways by just sort of forming a tunnel through the cell membrane, allowing larger or allowing different types of molecules to pass through. So when we have these transport proteins, then molecules can diffuse it. Okay. Other times sometimes it's sort of like a tunnel. Other times the molecules bind to the carrier protein. It changes shape and then allows them to go through. So it's another form of passive transport. Okay? But there are also proteins that allow for active transport, which again requires the use of energy to move molecules from a lower concentration to a higher concentration, to pump these molecules the opposite way. Okay? Just like if you have water, it's just going to run downhill. It's going to run downhill in a stream. If you're going to try and get that water back up to the top of the hill, you're going to have to exert some energy. You have to pump it up there using some energy. And if we look a little more closely at endocytosis or exocytosis, what do you think exocytosis is? Yeah, things moving out of the cell. Endocytosis, things coming into the cell. Okay? So penocytosis, we know the membrane indents or pinches in. A vacuole forms. And this is one way in which cells can take in the extracellular fluid. Phagocytosis, the cytoplasm streams and oozes around the molecule until it's fully ingested, and then it's in a vacuole.
Kind of like an amoeba?
Yeah, an amoeba, pseudopods work. That's an example of phagocytosis. Our white blood cells, some of our white blood cells work just like an amoeba. They find bacteria or other pathogens, and they ooze around them through phagocytosis. And then that molecules inside of the cell where it can be broken down by things like the lysosomes, which merge with that vacuole, and those enzymes break down whatever was in it, and that's how those things are broken down.
Like
Right.
how long does it take to like go around the cell and like digest it?
For the lysosome to digest, I don't know a time span. I would say a few minutes maybe, but I'm just sort of guessing. Exocytosis is sort of the opposite. It's when large molecules are going to leave the cell, and it works in the opposite way, where you have these bubbles of vesicles, for example, from the Golgi bodies, which go to the cell membrane, merge with it, and then sort of break open, allowing their contents to leave the cell, go into the extracellular fluid. An example of that is through neurotransmitters. So again, same things. Phagocytosis surrounding, pinocytosis pinching in, and then receptor-mediated, which requires certain molecules to bind to receptors before it pinches in. And again, here we see a neuron, a nerve cell. And in a nerve cell, these neurotransmitter chemicals, which are produced inside of the cell, are packaged up by the Golgi bodies into a little vesicle, which eventually makes its way to the outside of the cell, merges with it, breaks open, and releases these neurotransmitters into this extracellular space where it can then bind to the next neuron. And so that process is an example of exocytosis.
Hey, Mr. Dural,
Yes.
so when you get hurt, is that how like your nerves go to your brain and stuff?
Yeah, I mean, that's how messages are sent from different parts of our body through our nervous system. It works. So we have neurons, which are elongated cells, and a signal travels through an electrical signal down the neuron, but then it gets to the end. That neuron releases neurotransmitters. The next neuron picks them up, starts an electrical signal, brings it the other way. So it's this transfer of electrical signal, chemical, very quickly until that message gets to the brain or from the brain to your. That's how our nervous system works, through a combination of electrical and chemical signals. Any questions about that? All right. Nicolette?
Isn't there like a response to pain that doesn't travel to your brain because it would take too long?
Is there an example? I mean, like a reflex. You know, reflex is a message that doesn't go to your brain. It sort of arcs through your spinal cord and back to allow you to have a more sort of automated response to a stimulus. For example, yanking your hand back from something that's hot. That message goes from temperature receptors on your hand to your spinal cord and straight back out to your muscles, allowing you to pull your hand back very quickly. Okay, so you don't have to bring that message to your brain, interpret it, say, oh, this is really hot, I should move my hand, then send that message to move your hand back to your muscles. That would take significantly longer, and you would do more damage to your body. So instead you have that automatic. So if you ever burned yourself or something, you don't feel it at first. You yank your hand away, you don't think about it, and it starts to hurt, a few seconds later. So it's a protective mechanism.
So like if you didn't have like temperature or whatever in your hand, you would just keep it there.
Yeah, and you have to think, oh, this is really hot, this hurts, and then you would move it away, and by that time you would have done more damage to your hand. Yep.
Like if you're getting kicked in the face and then like think it doesn't hurt and then like 10 seconds later you're like down.
Yeah, it takes a little while for those messages to be sent. All right.